This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is a silent mutation? Answer: a silent mutation is a genetic change that codes for the same amino acid and often involves the third position of the codon. Question: what is a missense mutation? Answer: a missense mutation is a genetic change that results in a different amino acid. These mutations can be conservative if the new amino acid has a similar chemical structure. An example of this is sickle cell disease, which is a substitution of glutamic acid with valine. Question: what is a nonsense mutation? Answer: a nonsense mutation is a genetic change that results in an early stop codon, usually resulting in a non-functional protein. These mutations are commonly caused by UGA, UAA, UAG codons. Question: what are the different types of amino acid changes that can occur in a missense mutation? Answer: missense mutations can result in a different amino acid. These mutations can be conservative if the new amino acid has a similar chemical structure. It can also be non-conservative if the new amino acid has different chemical properties. Question: how are stop codons involved in nonsense mutation? Answer: nonsense mutations result in the creation of an early stop codon. This causes the translation process to stop prematurely, resulting in a non-functional protein. These mutations are commonly caused by UGA, UAA, UAG codons. Question: what is the impact of silent mutation on the protein? Answer: silent mutations have no impact on the protein because they code for the same amino acid. Since the amino acid sequence remains unchanged, the protein structure and function remains unchanged as well. Question: how do missense mutations affect the protein's function? Answer: missense mutations can have varying effects on the protein's function. If the new amino acid has a similar chemical structure to the original, the protein's function may remain relatively unchanged. However, if the new amino acid has different chemical properties, it can significantly affect the protein's structure and function. Question: how do nonsense mutations affect the protein's function? Answer: nonsense mutations result in a premature stop codon, causing the translation process to stop prematurely, resulting in a truncated, non-functional protein. This will cause a loss of function for the protein. This can be detrimental for the organism. Question: are silent mutations always harmless? Answer: although silent mutations do not change the amino acid sequence, they can still have an impact on the protein's function. They can change the Mr. NA stability, codon usage, and splicing, which can affect the protein stability, translation efficiency, and activity. They can also have an impact on the regulation of gene expression. Question: can missense mutations lead to genetic diseases? Answer: yes, missense mutations can lead to genetic diseases. They can cause a change in the protein structure and function, which can affect the normal biological processes of the organism. Examples of genetic diseases caused by missense mutations include cystic fibrosis, sickle cell anemia, and Tay-Sachs disease. Question: are nonsense mutations always harmful? Answer: yes, nonsense mutations are almost always harmful, as they result in a premature stop codon and a truncated, non-functional protein. This can cause a loss of function for the protein and can be detrimental for the organism. Some rare cases of nonsense mutations might not be harmful, but this is very rare. Question: can silent mutations be used as a marker in population genetics? Answer: yes, silent mutations can be used as a marker in population genetics. As they do not affect the protein's function, they are more likely to be inherited. This allows researchers to track the frequency and distribution of these mutations in a population over time. Silent mutations can also be used to infer evolutionary relationships between different populations. Question: how do missense mutations affect the protein stability? Answer: missense mutations can have a significant impact on the protein stability. They can cause changes in the protein structure, which can affect its ability to fold correctly and maintain its structure. This can lead to a decrease in the protein stability and increase the likelihood of the protein being degraded.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.